Hello pasangala, vanakkam and welcome. நான் தான் உங்க master teacher Nivetha, making bio learning very simplified just for all of you guys. So இன்னைக்கு செஷன்ல நம்ம பார்க்க போறது molecular basis of inheritance ஓட second part, correct? NCERT questions நேத்து ஒரு 88 questions பார்த்தோம். So இன்னைக்கு மிச்ச questions தான் பார்க்க போறோம், starting from the topic genetic code. So genetic code ல இருந்து தான் இன்னைக்கு நம்ம செஷன்ல நம்ம ஆரம்பிக்க போறோம். வணக்கம் and welcome to the session. A very good evening all of you pasangala. ரொம்ப முக்கியமான session. Okay, so session குள்ள போறதுக்கு முன்னாடி I have an important piece of information for all of you guys. So நம்ம Shiman sir உங்க ஊருக்கு வராரு, வாத்தி ride in Tamil Nadu. It's going to happen. So என்ன பண்ண போறாருன்னா, he's going to help you in career counseling pasangala. Parent awareness session நடக்க போகுது. He's going to help you in trying understand the various career aspects. Okay, and you can also avail exclusive offers and scholarship on Vedantu courses. So இது எப்பத்துல இருந்து எப்ப நடக்க போகுதுன்னா 10 AM ல இருந்து 4 PM வரைக்கும் நடக்க போகுது. Okay, March 14th இன்னைக்கு, today was March 14th. Okay, நாளைக்கு திருச்சி, then 16th Chennai, 17th Velachery Chennai, 19th Coimbatore, 21st Kovilpatti, 23rd Tambaram, 24th Avadi. Okay, so this is what is the schedule pasangla. Okay, why are you guys still waiting? Go get yourself registered. All the details is given in the link. Okay, ஒரு descriptionல நாங்க link கொடுத்திருக்கோம், அதுல போய் நீங்க register பண்ணுங்க. register பண்றது ரொம்ப முக்கியமான விஷயம். Okay, where are all of you pasangla? Come on, a very important session. NCERT questions, this is very important. Hi, good evening. Okay, start பண்ணிரலாமா? Time waste பண்ண வேண்டாம். எவ்வளவு சீக்கிரம் முடியுதோ அவ்வளவு சீக்கிரம் ஆன்சர்ஸ் பண்ணலாம். A very good evening all of you. So இது நம்ம நேத்தே டிஸ்கஸ் பண்ணிருந்தோம். We had discussed this earlier. Okay, 2021 ல 9 question, 2022 ல 7 question, 2023 ல 7 question கேட்டிருந்தாங்க. Hi everyone, good evening. எல்லாரும் எப்படி இருக்கீங்க? How are you guys? Yes, super. Shall we start the session without wasting even a minute? Yes, sharp 8:1. Okay, we will start the session. Happy good evening. Yes, good evening. Yeah, start பண்ணிரலாம் session பசங்களா, இன்னும் டைம் வேஸ்ட் பண்ண வேண்டாம். ஸோ நேற்று எது வரைக்கும் பார்த்தோம்னா, we spoke about transcription வரைக்கும் பார்த்தோம். இன்னைக்கு டிரான்ஸ்லேஷன் ஸ்டார்ட் பண்ணிடலாம். அதே நம்பர் தான் போட்டிருக்கேன், 88லேருந்து ஸ்டார்ட் பண்ணியிருக்கேன். ஓகே, டியூரிங் ரெப்ளிகேஷன் அண்ட் டிரான்ஸ்கிரிப்ஷன். ஓகே, நான் ரொம்ப ஃபைன் தான். ஆக்சுவலாக ரொம்ப டயர்டாக இருக்கேன், வெளியே காமிச்சிக்கல. ஓகே, தட்ஸ் ஹவு த எனர்ஜி ஷுட் பி. ஓகே, ஸோ அதனால தான் கொஞ்சம் எனர்ஜியாக இருக்க மாதிரி நம்மளை பார்த்துக்கணும். தட்ஸ் இட். ஓகே, yeah. So during replication and transcription, a nucleic acid was copied from another dash. Okay, during replication and transcription, a nucleic acid was copied from another dash. Who will tell me? NCERT line. I saw so many things today while preparing these questions. Okay, pudu vinavana questions kandipa keklam. Very good. The right answer is nucleic acid. Okay, so inga pathingana transcription nalu seri, replication nalu seri, they are produced from nucleic acid. Super. The process of translation requires transfer of genetic material from a polymer of dash to synthesize a polymer of dash. Understand the question. Okay, so இந்த லெசனே படிக்காதவங்க தயவு செஞ்சு உங்க NCERT புக்கை திறந்து ஆன்சர் சொல்லலாம், பரவாயில்ல. ஓப்பன் புக் டெஸ்ட். ஓகே, எஸ், டெல் மீ த ஆன்சர் பசங்களா. The process of translation requires the transfer of genetic information from a polymer of dash to dash. Okay, it's from a polymer of nucleotides, correct, to synthesize a polymer of amino acids. So amino acids produce பண்ணப்படுது. Super, very good. It was Dash, a physicist, who argued that since there are only 4 bases and if they have to be code for dash, the code should constitute a combination of bases. கொஞ்சம் confusing. யாருப்பா இந்த scientist? Who is this scientist? Anybody knows? It starts with the letter G. Anybody knows the scientist name? It is George Gamow. Okay, it is George Gamow. So அவர் வந்து ஆர்கியூ பண்ணாரு. ஓகே, இத்தனை கோடான் தான் இருக்கணும்னு சொல்லி. ஓகே, because there are only 20 amino acid, the code should constitute a combination of bases. Okay, George Gamow, please do understand. இதெல்லாம் இது வரைக்கும் கேட்கல, இனிமேல கேட்க வாய்ப்புகள் அதிகமா இருக்கு. Okay, super, super. அடுத்த கேள்விக்கு போகலாம். He suggested, யாரு? George. Okay, he suggested that in order to code for all the 20 amino acid, the code should be made up of dash nucleotide. This was very bold proposition because a permutation combination of dash would generate dash codons. This is very, very interesting. Okay, he told that it is made up of 3 nucleotide. Okay, a permutation combination 4 into 4 into 4, that is 4 cube, okay, would generate how many codon? 64 codons. Very, very, very important. Yes. Super, super. Okay, எத்தனை codon நம்மகிட்ட இருக்கு? 64 codon இருக்கு. Yes, very good, very good. Next question. The chemical method by dash was instrumental in synthesizing dash molecule with defined combination of bases. Who developed the chemical method? Yes. Who developed the chemical method? What's the answer, pasangla? Inga enna answer varudhu? Hargobind Khorana, famous Indian scientist. Okay, Hargobind Khorana. Corona mari Khorana ஞாபகம் வச்சுக்கோங்க. Okay. So Hargobind Khorana, he was a famous Indian scientist, okay. So was instrumental in synthesizing dash molecules with defined bases. Inga enna pa? What synthesizing dash molecules? Synthesizing RNA molecules, okay, with defined combination of bases. With defined combination of bases. ரொம்ப ரொம்ப முக்கியம் இதெல்லாம். Okay, இது வரைக்கும் கேட்கல, இனிமேல கேட்கலாம். இனிமேல கேட்கக்கூடிய சான்ஸ் அதிகமா இருக்கு. Dash cell-free system for protein synthesis finally helped the code to be deciphered. யாரோட? இங்க ஒரு சயின்டிஸ்ட் பேர் தான். இங்க ஒரு சயின்டிஸ்ட் பேர் தான். Who will tell me the scientist name? Here it is Marshall, okay, Marshall Nirenberg. Marshall Nirenberg. Okay, cell-free system for protein synthesis finally helped the code to be deciphered. So இங்க வந்து மூணு சயின்டிஸ்ட் வராங்க. ஒன்னு வந்து ஜார்ஜ், இன்னொன்னு வந்து ஹர்கோபிந்த் கொரானா, இன்னொன்னு வந்து மார்ஷல் நிரன்பர்க். ரொம்ப ரொம்ப முக்கியம். Okay, very, very important. கூடி சீக்கிரம் கிளாஸ் 12க்கு தனியா சயின்டிஸ்ட் பேரு, கிளாஸ் 11க்கு தனியா சயின்டிஸ்ட் பேரு போடுறேன். Okay, ஒரு ரெக்கார்டிங்கா உங்களுக்காக போடுறேன். Yes, next question. The codon is dash. Okay, dash codons code for amino acid and dash codons do not code for amino acid, hence they function as dash. These are all coming under properties. Properties. Yes. எங்க இருக்கு அது? Where is it in your textbook? Okay, they have given it is a polynucleotide phosphorylase. It's an enzyme. It is an enzyme. Okay, அது வந்து ஒரு enzyme. Okay va? Yeah, so the codon is dash, the codon is triplet. The codon is what? Triplet, okay. Dash codons code for amino acid, 61 codons code for amino acid, okay. And 3 codons do not code for amino acid, hence they function as terminator codons, okay. Terminator codons. ரொம்ப 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 முக்கியமான விஷயம். Okay, so phosphorylase removes the phosphate group. Okay, phosphatase, phosphorylase, they all remove the phosphate group. Okay, yes. So some amino acid are coded by more than one codon, hence terminator illa stop மூணுமே ஒண்ணுதான். So இந்த terminator codonக்கு இன்னும் ரெண்டு பேர் இருக்கு, stop codon or nonsense codon. எல்லாமே ஒரே விஷயம் தான். Okay, yes. Next information. Some amino acids are coded by more than one codon, hence the codon is degenerate. ரொம்ப ரொம்ப முக்கியம். Okay, degenerate or degeneracy of codon. Okay, yes, very good, very good. The codon in mRNA is read in a dash. There are no dash. It is read in a dash manner, pasangla. Who will tell me? Very easy properties of genetic code. It is read in a contiguous fashion. Okay, contiguous fashion, GUOS, okay, fashion. There are no punctuation marks or there are no punctuations. Okay, yes, contiguous fashion. Super, super. Okay, அடுத்தது the codon is nearly dash. Every line is so very important. The codon is nearly universal. Okay, for example, from bacteria to human, dash would code for phenylalanine. Who will tell me this? Dash should code for phenylalanine. Am I going in the right speed? Am I too slow? Am I too fast? Please do tell me. Yes, please do let me know. Dash would code for phenylalanine. Quick-ஆ சொல்லுங்க பசங்களா. Super, super. You would code for phenylalanine. Okay, some exceptions to this rule would have been found in dash and dash. Where do you find these exceptions? We find these exceptions in mitochondria. Okay, இதையும் கேட்கலாம் statement based questionல மாத்தி கொடுத்துடலாம். Okay, and some protozoans. இது வந்து சில மைட்டோகாண்ட்ரியா அப்புறம் புரோட்டோசோவன்ஸ்ல மாறி இருக்க வாய்ப்புகள் அதிகமா இருக்கு. Okay, am I clear? Yes. Can we continue? Okay, AUG has dash function. It codes for dash and it also acts as dash. AUG has dash function. It codes for dash and it also acts as dash. Come on பசங்களா, quick-ஆ சொல்லுங்க. AUG has a dual function. அதுக்கு வந்து ரெண்டு function இருக்கு. Okay, it codes for methionine. Okay, it codes for methionine and it also acts as an initiator codon. அதுக்கு வந்து ஒரு dual function இருக்கு. ஒரு லைனுமே possible question. Every line is possible question. Okay, yes, super. It is a start codon இல்ல initiator codon. ரெண்டுமே absolutely correct. Okay, dash are the terminator codons. Which are all the terminator codons? எதெல்லாம் பா terminator codons? யார் சொல்றீங்க? UAA, UAG, UGA. They are all your terminator codon. You should never ever forget this. Okay, dash may result in, we are slowly moving on to the next topic. Very good, very good. Dash may result in loss or gain of a gene and so a function. Dash may result in loss or gain of a gene, so a function. சொல்லுங்க, ஒரு ஃபங்க்ஷனே லூஸ் பண்ணக்கூடிய ability யாருக்கு இருக்கு? Who is having it? It is mutation. Okay, so mutation may result in loss or gain of a gene and so a function. Okay, very important. So mutation na enna? It is a sudden inheritable change in the DNA. Okay, next, a classical example of point mutation is change of dash in the gene for dash chain that results in the change of amino acid residue from dash to dash. Sickle cell anemia. Who will tell me this? Who will tell me this? What is changing to what? What is changing to what? It is a change of single base pair. Okay, in the gene for what? Beta globin. Okay, beta globin. It changes the amino acid from dash to dash. Amino acid is changed from what pasangla? Glutamate to valine. It is so very important. This has already been a PYQ. இது நிறைய வாட்டி PYQ already கேட்டிருக்காங்க. Okay, it results into a diseased condition called sickle, okay, cell anemia. Okay, it results in a condition called what pasangla? Sickle cell anemia. Very very important. Very important pasangla இந்த ரெண்டு லைன். So இது என்ன மாதிரி mutation ன்னு சொல்றோம்? It's called point mutation. So what is this kind of mutation called? Point mutation is change in the single base pair is what we call it as point mutation. புரிஞ்சதா? எல்லாரும் glutamate to valine ன்னு போடுங்க chat sectionல. ஏன்னா நிறைய பேர் அது valine to glutamate ன்னு இன்னும் confuse ஆயிட்டு இருக்கீங்க. Okay, yes. Shall we continue? Yes. Insertion or deletion of one or two base pairs changes the dash from the point of dash or dash. Okay, we are slowly moving on. We are slowly moving on. Okay. Yes. Yes. The insertion or deletion of one or two base pairs. Very good. The insertion or deletion of one or two base pair changes the reading frame. Okay, reading frame from the point of insertion from the point of insertion or deletion. Okay, so ஒரு nucleotide ஒரு base pair இல்ல ஒரு insertion இல்ல deletion நடந்தா the entire reading frame is changed. So அத நாம என்னன்னு சொல்றோம்? Frame shift mutation ன்னு சொல்றோம். Okay, so we call it as frame shift. Okay, frame shift or deletion mutation. என்னன்னு சொல்றோம் பசங்களா? Frame shift or deletion mutation ன்னு சொல்றோம். ரொம்ப 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 முக்கியம். Okay, I am not joking. All these are possible questions. இதே தவிர வேற ஒண்ணுமே கேட்க முடியாதாங்க. Okay, dash postulated. Coming to tRNA, we are slowly moving on to tRNA. Good evening, doctor said ரொம்ப நாள் ஆச்சு படிக்கிறதுல ரொம்ப பிஸியா இருக்கீங்களா? Good. Yes. Frame shift, deletion, mutation. Super. இப்போ சொல்லுங்க பசங்களா. Dash postulated the presence of an adapter molecule that would on the other hand read the dash on the other hand would bind to specific dash. Can anyone tell me? Very good. Super. Crazy dude. It was Crick. So Crick வந்து மூணு விஷயம் சொல்லியிருக்காரு. Francis Crick மூணு விஷயம் சொல்லியிருக்காரு. யாராவது சொல்றீங்களா? What are the three important things that Crick proposed? Crick proposed double helix model of DNA. Crick proposed central dogma of molecular biology. Crick proposed the presence of tRNA molecule. Okay, இந்த மூணு விஷயத்தை வந்து அவர் டிஸ்கவர் பண்ணாரு. Okay, postulated the presence of an adapter molecule that would on one hand read the dash, on the other hand would bind to the specific dash. Who will tell me this? Would read the code. Okay, it would read the code and it also be able to bind to specific amino acid. It would read the code, okay, and would also bind to specific amino acid. Okay, புரிஞ்சதா? எல்லாருக்கும் புரிஞ்சதா? Yes, very important. So என்னோட tRNA molecule ஒரு part of my tRNA that if imagine this is my anticodon, it goes and binds to codon. Okay, and this will go and bind to your, where this will go and bind பசங்களா? Amino acid. Okay, so quiz questions கண்டிப்பா போடலாம். இந்த NCERT question NEET பசங்களுக்கு ரொம்ப யூஸ்ஃபுல்லா இருக்கும். Okay, அதனால தான் especially in this chapter molecular basis, so அதனால தான் இத பண்ணிட்டு இருக்கேன். Okay, yes. Okay. So how was the test going on? எப்படி டெஸ்ட் நல்லா இருந்துச்சா? The tRNA then called, அப்போ அந்த tRNA என்னன்னு சொல்லிட்டு இருந்தாங்க? Can anyone tell me what is a tRNA called? Very good. It is called soluble RNA. So இது வந்து உங்க NCERT ஃபுல்லா ரிவைஸ் பண்ணிரலாம். Okay, MCQs போடுறேன். Important MCQs from molecular basis of inheritance கண்டிப்பா போடுறேன் இந்த வீக்லயே. Okay, was known before the genetic code was postulated. Very good. sRNA. Yeah, tRNA has a dash loop that base is complementary to the dash. tRNA has a dash loop. Very simple. Okay, it has an anticodon loop. Okay, it has an anticodon loop that base is complementary with the codon. Okay, codon, anticodon will base pair with codon. Okay, tRNA also has a dash end to which it binds to dash. tRNA also has what pasangla? What does a tRNA has? Anybody? tRNA also has an amino acid acceptor end. Okay, acceptor end to which the amino acid binds to, which it binds to amino acid. Yes, acceptor end. Okay, acceptor end. Let's make it very simple. Okay, yes, amino acid acceptor end. Super. tRNAs are specific for each dash. They are specific for each amino acid. Okay, ரொம்ப முக்கியம். For initiation, there is a specific tRNA. It is referred to as dash. There are no tRNAs for dash. Very interesting question. Who will tell me this? Yes. For initiation, obviously you have an initiator tRNA. என்ன இருக்கு? Initiator tRNA இருக்கு. கரெக்ட்டா? So there are no tRNAs for dash. எதுக்கு வந்து tRNAs இருக்காது? Can anyone tell me? Very good. Super. Okay, there are no tRNAs for stop codons. Stop codons க்கு tRNA வே கிடையாது. Okay, initiator tRNA என்ன amino acid carry பண்ணும்? Initiator tRNA எந்த amino acid carry பண்ணும்? Who will tell me? Who will tell me initiator tRNA எந்த amino acid carry பண்ணும்? Any idea? Any idea? Initiator tRNA obviously carries methionine. அது என்ன amino acid carry பண்ணும்? Methionine amino acid carry பண்ணும். Okay, super, super. Many of you are telling it right. அடுத்த கேள்விக்கு போகலாம். The secondary structure of tRNA has been depicted that looks like a dash. In actual structure, tRNA is a compact molecule which looks like dash. Interesting question, very important question. They have asked this question. Who will tell me the answer? Yes, so initially it looks like a clover leaf. Okay, so idu vandu, but in an actual structure it looks like inverted L. Idu ketirukanga pasanga, inverted L. Okay, romba mukkiyam, very important. It's a compact molecule which looks like inverted L. Okay, yes. Shall we continue? Shall we continue pasangla? Yes. Translation refers to the process of polymerization of amino acid to form a dash. Polymerization of amino acid edukku nadakudhu? Very simple, very easy to form a polypeptide chain. To form a polypeptide chain. Romba mukkiyamaana vishayam. Okay, protein, polypeptide, rendume correctana answer da. Okay, the order and sequence of amino acid are defined by the sequence of bases in the dash. Interesting question. Interesting question. The order and sequence of amino acid are defined by the sequence in dash. Yes, yes. Sequence of the bases in the mRNA. Obviously. Okay, the amino acids are joined by a bond which is known as. இதெல்லாம் ரொம்ப easy question. ரொம்ப easy question. எல்லாருமே சொல்லணும். That is what we call it as codon. Absolutely correct. Very good. இதுக்கான ஆன்சர் என்ன? Peptide bond. How is a peptide bond formed? Anybody? How is a peptide bond formed here? Yes. Therefore, in the first phase, என்னோட translationல நடக்கிற first phaseல amino acids are activated in the presence of dash and linked to their dash. Okay, how are the amino acids activated? How are the amino acids activated? They are activated in the presence of ATP. Okay, and they are linked to their cognate amino acid. Okay, a process commonly called as, enna solrom indha process? Charging of, okay, charging of tRNA or activation nu solroma. Is it called activation? Yes, amino acylation. Activation illa amino acylation. Okay, we call it as amino acylation. Okay, oru லைனுமே ரொம்ப முக்கியம். Yes, charging nu solrom illa amino acylation nu solrom. Very good. Okay, acylation means transfer of amino group. Okay, yes. அடுத்தது, the cellular factory responsible for synthesis of protein. Where is the entire protein synthesized? சின்ன வயசுல இருந்து படிச்சிட்டு வரோம். protein வந்து எங்க synthesize பண்ணப்படுது? Obviously ribosome. Okay, the more faster you go, the faster we can finish. The ribosome consists of dash and about dash protein. Yes, the ribosome consists of dash and about dash protein. The ribosome consists of RNA and inga or protein number super. It consists of 80 different proteins. Okay, in its inactive state, it exists as two subunit. Rombo easy. A larger subunit, okay, and a smaller subunit. Okay, the ribosome acts as a catalyst. Okay, what is that catalyst called? Ribozyme. Okay, adoda per enna? What is the name of that catalyst? Can anyone remember? Any ribozyme? It is 23S rRNA. Okay, this 23S rRNA has a peptidyl transferase activity. Abdina enna ma'am? It helps in formation of peptide bond. 23S rRNA எல்லாருமே chat section la போடுங்க. Very, very important. Okay, 23S, not 18S. 23S is involved in peptide bond formation. Okay, yes, in the formation of peptide bond. Okay, can we continue? Can we continue? Yes. A translation unit in mRNA is a sequence of RNA that is flanked by the dash and the dash codes for a polypeptide. So, this is our translation unit. In the endliyu enna irukonu kekranga. What codes will be there? They are asking. What are the codes? Anybody? Anybody? It has an initiator. Okay, initiator. So, you have initiator and the terminator. Okay? Yes, initiator and terminator. Yes. Okay. Okay, sure. Okay, இது difficult-ஆ இருந்தாலும் இதுதான் உங்களுக்கு exam-க்கு help பண்ணும். Okay, this is what is going to help you in exam. கொஞ்சம் ஒரு நாள் ரெண்டு நாள் கஷ்டமா இருக்கும். Okay, but later on you will get used. I am going to arrange MCQs, I am going to do week quiz. Okay, but this is the best method of studying, trust me. Okay, yeah. So an mRNA also has some additional sequence that are non-translated and are referred to as UTR, untranslated region. Okay, so the UTRs are present at both the ends. Okay, that is called 5' UTR, that is before the start codon. In the oru line romba mukkiyam. Okay, and at the 3' end, okay, they are required for efficient, correct? They are required for efficient translation process. They are required for efficient translation process. Okay, purinjada pasangala idu varaikum? Next, for initiation, the ribosome binds to the dash at the start codon, dash that is recognized only by the dash. Who will tell me? For initiation, ribosome binds to the mRNA, obviously, okay, to the mRNA at the start codon, okay, dash that is recognized only, efficiency, to improve efficiency. Very good, very good. Okay, yes, that is recognized by what? Yes, who recognizes that? Your initiator tRNA, iTRNA. Okay, that is recognized only by the at the start codon AUG, correct? Inga da idu varanum. Okay, yes, at the start codon AUG, okay, that is recognized by iTRNA. Yes. Okay, translation I am talking about. Yes. Okay, yeah. Pogalama, can we continue? Can we continue, all of you? Any doubts? Any doubts here? Yeah, we will continue. Yes. Next question. The ribosome move from codon to codon along the dash. Ribosome eppadi move agum? Oru codon la irundhu innor codon move agum. Eppadi move agum? Along the mRNA. Okay, along the mRNA. Amino acids are added by one, translated into a polypeptide sequences, dictated by dash and represented by dash. Very interesting question. This is a very interesting question. Okay, who is dictating it? Who is directing it? Who is representing it? Okay, dictated by DNA, represented by RNA. Okay, so you all know DNA becomes mRNA, mRNA becomes protein. எவ்வளோ grammar இருக்கு பாருங்க, எவ்வளோ vocabulary இருக்கு பாருங்க, ஒரு வேர்டுமே ரொம்ப முக்கியம். Okay, yes, all of you clear? எல்லாருக்கும் புரிஞ்சதா? Can we continue? Represented by mRNA, let's put it like that. Yes. Okay, at the end a dash binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. At the end a dash binds. What binds? Super, a release factor. So, இப்படி நீங்க படிச்சாலாம் 360 out of 360 வாங்க முடியும் பசங்களா. ஒரு லைனை கூட விடக்கூடாது. ஓகே, therefore ரோ ஃபேக்டர் வந்து டிரான்ஸ்கிரிப்ஷன், ரிலீஸ் ஃபேக்டர் வந்து டிரான்ஸ்லேஷன். ஓகே, yes, therefore in simple terms it is the dash dash or dash conditions that regulate the expression of genes. We are slowly moving on to regulation of gene expression. சொல்லுங்க பசங்களா, this is also there in your textbook. This is also there in your textbook. I think it is a physiological, okay, and morphological conditions. மூணு condition இருக்கு. யார் என்ன கரெக்ட்டா சொல்றீங்க? Therefore this comes to regulation. Okay, in regulation we are talking about, who will tell me the right answer? Very good, very good. It is a transcriptional, metabolic and physiological, correct? Correct? So it is the metabolic, physiological, idu ஞாபகம் இருக்கு எனக்கு. Okay, physiological or transcriptional. Okay, environmental, super, super. Physiological, metabolical and environmental, super, super. So இத வச்சு ஒரு கேள்வி ஃபார்ம் பண்ணலாம். ஏன் விடணும்? ஓகே, ஏன் நம்ம இதை விடணும்? நம்ம டைம் இருக்கும்போது, நம்மளால முடியும் போது ஏன் இதை நம்ம விடணும்? கண்டிப்பா படிக்கணும். ஓகே, the dash and dash of embryo into adult organism are also a result of the coordinated regulation of expression of several sets of gene. The development and differentiation, yes, the development and differentiation, DVPT na development, okay, and differentiation, okay, of embryo into adult. Okay, yes, that is also a result of gene regulation. இப்போ நம்ம லாக்-ஓபெரான் குள்ள போறோம் slowly. Okay, yeah, shall we get into லாக்-ஓபெரான்? Shall we get into லாக்-ஓபெரான்? Are you all ready pasangla? Shall we continue? Can I get some thumbs up in the chat section? Yes. The accessibility of the promoter region of prokaryotic DNA, as in many cases, regulated by the interaction of protein with specific sequence. Where does this protein go and interact? Where does this regulatory protein go and interact? One important region. Who will tell me? It starts with the letter O. It starts with the letter O. Yes. Where is it going to interact? Solite. It starts with the letter O. Yes, little bit slow. Sure. Konja slow pandra. Yes. Board question, not sure. Board la indha mari kekka mattanga, but direct fill in the blanks idhula irundhu kekkalam. Direct fill in the blanks idhula irundhu kekkalam. It is operator. Operon illa, operator. So the regulatory protein enga poi bind agum sollirukanga. It is operator. Okay, dash, the first to elucidate a transcriptionally regulated system. Who basically discovered this transcriptionally regulated system? Anybody? Rombo famous ana rendu per, Jacob and Monod. Yarpa? Jacob and Monod were the one who first elucidated the transcriptionally regulated system. They were awarded Nobel Prize for this. Okay, can we continue? Can we continue? In லாக்-ஓபெரான், a dash structural gene is regulated by a common dash and dash genes. In லாக்-ஓபெரான், a polycistronic, okay, எப்படி இருக்கு structural gene? A polycistronic, okay, a polycistronic structural gene is regulated by a common dash and dash genes. What is polycistronic? They have multiple structural genes, correct? Yes. What is regulated by? Very good, very good. Adha answer. Who is regulating this structural gene? Yes, yes. Very good, very good. And the I gene enna protein produce pannom? Gandhi, can you tell me? Can you tell me? Yes, it is your I, that is your regulatory, okay, and operator region. Okay, yaar and idu control pandra? Regulatory and operatory region. Promoter and regulatory ya? Repressor protein, regulatory protein, rendume correct da. Okay, yes, yes. Can we continue? Very good, super. Yes, such arrangement is very common in bacteria and is referred to as, ipo solunga answer, it is referred to as operon. Okay, there are so many operons pasangala, எக்கச்சக்கமான operons இருக்கு. Okay, so you have lac operon, you have trip operon, okay, lac operon, trip operon, okay, arabinose operon, நிறைய operons இருக்கு, right? operon க்கு operator க்கு confuse ஆகாதீங்க. Yes, veline, arabinose, a lot of it is there. Veline, arabinose. So நம்ம படிக்க போறது வெறும் lac operon தான். Okay, we are going to study only lac operon. Okay, is this speed okay? Neetu in the speed okay va? Yes, very good, very good. The lac operon consists of dash regulatory gene and dash structural gene. So lac operon la எப்படிப்பட்ட regulatory gene இருக்கு? Yes, one regulatory gene. Need more speed? Okay, speed கம்மி பண்ண சொன்னாங்க, so I reduced. Okay, I will be a little more fast. இதெல்லாம் easy. It has one regulatory gene and it has 3 structural gene. Okay, the I gene codes for the dash of the lac operon. The I gene codes for the dash of the lac operon. Yes, 132. The I gene codes for the dash of the lac operon. Very easy. It codes for the repressor of the lac operon. The repressor anu சொல்லுவாங்க, இல்ல regulatory protein னு சொல்லுவாங்க. ரெண்டுமே ஒரே விஷயம் தான், கன்ஃபியூஸ் ஆகாதீங்க. Okay, yes, super, super. The Z gene codes for dash. இங்கே இருக்கு answer. Okay, it codes for, நான் எழுதுறதுக்கு முன்னாடி எழுதுங்க பாப்போம், beta galactosidase. The Z gene codes for beta galactosidase. Okay, it is responsible for the hydrolysis of the disaccharide. What is a disaccharide? Lactose. Very good, very good. Unknown எனக்கு முன்னாடி முடிச்சிட்டீங்க. Okay, into glucose and galactose. Okay, this line is so very important, super. Okay, the Y gene codes for permease, which increases the permeability of the cell. Permeability na enna? It allows certain substance to come inside. That is what we call permeability. So inga edhupa permeability? Lac operon capping and tailing? No, lac operon is a prokaryotic gene. Adla capping tailing ella nadakadu. Veru eukaryotes la mattum da capping tailing ella nadakum. Yes, very good. To allow lactose. Okay, next set of questions. The gene encodes, the A gene, already answer kuduthutta. The A gene encodes for transacetylase. It helps in acetylation reaction. Okay, hence all the 3 gene products in lac operon are required for the metabolism of lactose. Obviously. Okay, dash is the substrate for the enzyme dash and it regulates switching on and off of the operon. Hence it is termed as dash. Yes, question number 137. So dash is the substrate, lactose is the substrate. Okay, for the enzyme what? Beta galactosidase. Okay, hence it is termed as, anyone can fill this for me? Anyone can fill this for me? Yes, anyone can fill this? Hence it is termed as, what do we call? Super, Neetu, super. Hence we call it as inducer. Adha enna solrom? Inducer nu solrom. Super. Okay, the repressor of the operon is synthesized all the time constitutively from the dash. From where are these repressors secreted? From the laci. So idhula enna puriyuduna, idha clear ah purinjukonga. Constitutive oda meaning purinjukonga. It is always on. எப்போமே இந்த repressor protein இருந்துட்டே இருக்கும். Okay, 134 allows lactose. Okay, yes. So 138 done. எல்லாருக்கும் புரிஞ்சுதா? lac I இல்ல I gene ன்னு கூட எழுதலாம். Super. Yes, we shall continue. அடுத்த question, there are few more questions. இன்னும் 2 தான் இருக்கு. Human genome project, அப்புறமா DNA fingerprinting. Okay, the repressor binds to the dash region of the operon and prevents RNA polymerase from transcribing the operon. Yes, from transcribing the operon. It binds to the operator. ரொம்ப முக்கியமான விஷயம். Okay, in presence of inducer such as dash and dash, ஒன்னு வந்து எல்லாருக்குமே தெரியும், lactose. இன்னொரு inducer பேர் என்ன? What is other name of inducer? when negative regulation okay 134 explain பண்ணணுமா ஒரு நிமிஷம் the Y gene codes for permease. Okay, so permease இன்றது ஒரு enzyme. இந்த enzyme என்ன பண்ணோம்னா it makes my bacterial cell more permeable. Okay, என்ன பண்ணோம் more permeable to lactose. So வெளியே இருக்க lactose அதிகமான lactose என்னோட செல்ல enter ஆகும். That is what we call it as increase in permeability. புரிஞ்சுதா? Who asked that question? I think Dr. M, right? Dr. M asked, right? Clear? Yeah. So it is lactose or allolactose. So what is this lactose and allolactose? They are isomeric forms. Avanga yaru pasangala? They are all isomeric forms. Okay? Yes. Very good. Very good. Yeah. The repressor is dash by interaction with the dash. The repressor is inactivated. Okay? The repressor is inactivated. Okay, உங்க எல்லாருக்குமே லாக் ஓப்பரான் இப்ப கண்ணு முன்னாடி வரணும். Okay, inactivated or inhibited by the interaction with the inducer. Okay, எல்லாருக்குமே லாக் ஓப்பரான் கண்ணு முன்னாடி வரணும். எத்தனை பேருக்கு வருது லாக் ஓப்பரான் கண்ணு முன்னாடி when you are doing this? Okay, regulation of lac operon by repressor is referred to as negative regulation. Okay, இந்த lac operon ஒரு சின்ன ஒரு minute detail சொல்லிடுறேன். switch off ஆகும்போது அது negative, switch on ஆகும்போது அது positive. So this operon is an example for both positive and negative. Simple. Okay, it's under the control of positive. இங்க negative இருக்கு, அப்போ positive regulation as well. Simple. Okay, yes. Yeah, positive is beyond this book. அங்கே கொடுத்திருக்காங்க textbook லே. Okay, now let's move on to human genome project. அசால்ட்டா ஆன்சர் பண்ணக்கூடிய ஒரு விஷயம். Okay, yes, எல்லாம் மெமரியில இருந்து எடுத்து போடணும், யோசிக்கவே வேண்டாம். Human genome project is said to have approximately dash, and if the cost sequencing is $3 per base, okay, the total estimated cost of the project would be approximately, yes, what is the total cost of the project? Very good, very good, very good. You have 3 billion base pair, that's a human genome. Very good. What is the approximated cost? Very good, 9 billion US dollars. That's a lot of money. Okay, அடுத்தது, the human genome project was a dash project coordinated by the US Department of Energy and National Institute of Health. Who will tell me this? Ecosystem? Sure, we will do it. Okay, it was a 13-year-old project. So 13-year project, okay, which was coordinated by mega project. Okay, 13-year project, anything. 13 years is in your NCERT, so we will follow this. 13 வருஷம் ஆச்சு. Okay, they started it in the year 1990. They completed it in the year 2003. Okay, one approach focused on identifying all the genes that are expressed as dash. So ரெண்டு approach யூஸ் பண்ணாங்க, ரெண்டு techniques யூஸ் பண்ணாங்க. அது என்ன techniques ன்னு கேக்குறாங்க. Okay, yes, 4 countries. Very good. What is the first technique? Anybody? Okay, the first technique was EST. Okay, very good, EST. The other technique was sequence annotation. என்னன்னு சொல்றோம் பசங்களா? அத sequence annotation ன்னு சொல்றோம். What is EST? Express sequence tag. இத மட்டும் மறந்துடாதீங்க. These two is important. They have asked it as a PYQ. நிறைய வாட்டி கேட்டிருக்காங்க. Okay, அடுத்த விஷயம் பார்க்கலாம். The commonly used, இதெல்லாம் தூக்கத்துல எழுப்பி கேட்டாலும் நீங்கலாம் சொல்லணும். Human genome projectல என்ன vectors யூஸ் பண்ணாங்க சொல்லுங்க, எல்லாரும் சொல்லுங்க. What are the vectors that was used in HGP, Human Genome Project? I want everyone to tell. எல்லாரும் சொல்லணும். Yes, ஏன் back end yak யூஸ் பண்ணாங்க? Why did they use back end yak? Why not plasmid? Why not bacteriophage? Why did they use back end yak? Any idea why did they use back end yak? Yes, எதுக்குப்பா back end yak யூஸ் பண்ணாங்க? Because back end yak could carry a huge size genome. பெரிய DNA fragments carry பண்ணக்கூடிய ability இருந்துச்சு. Okay, their insert size was high. Okay, copy number also, but primary thing is they could insert a huge DNA into it. Yes, very good. The fragments were sequenced using automated DNA sequences that worked on the principle of a method developed by Tirpi, scientist pair. Here we have a scientist name. Yes. Yes, here we have a scientist name. Can anyone tell me? Can anyone tell me? Sanger. Okay, Sanger sequencing and so more. Frederick Sanger. Okay. Yes. Clear? All of you? Frederick Sanger. Very good. Kandipa na scientist name alla or video potta and the PDF ungalkaga kudukren. Okay. Yes, Frederick Sanger. The sequence of chromosome 1 was completed only in eppo on the chromosome 1 oda sequencing complete pannanga. In which year? In which year did they complete? Anybody? Yes, it was completed only in the year May 2006. In the NEET, kandipa silavati idalam kekkalam. And the Earth Summit, okay, World Summit, adamari idiyum kekkalam. Okay, adaye vidanum, kandipa padichiralam. Okay, adutha vishayam, the human genome contains dash million base pairs. The human genome contains, coming to the salient features, okay. Yes, 2006 la da sequencing mudichanga chromosome 1 odadu. Yes. How many base pair does it contain? How many million? It is 3164.7. 3164.7. All of you put it in the chat section. We don't want to lose any marks. Yes. Very good. The average gene consists of dash bases. The average gene consists of 3000 bases. Idella neenga knowledge use pannom thevilla. Neenga remember by heart pannirukadha adu appadiye neenga inga sollanum. That's it. Okay? Yes. Yes, very good, super. Oh, which one? This one? Edhu solringa? Okay, so with the largest human gene, which is the largest human gene, pasangla, this is important. This is so very important. Which is the largest human gene? It is your dystrophin, the largest human gene. Okay, very good. It contains 2.4 million base pairs. Okay, yes. The total number of genes is estimated at total number of gene 30,000. So solli kuduthu enakku adu abbe mind la stick aichi. Okay, yeah, 30,000 genes are there. So initially they thought we have 80,000 to 1,40,000 genes. Okay, almost dash nucleotide base are exactly same in people. This is also very important. Evlo base pair almost same. Neenglu nanu evlo percent same. We are all 99.9% same. Okay, 99.9% same. Yes, very good, super. Okay, adutha vishayam paakalama. Shall we try to understand the next question? The function are unknown for over 50% of the discovered genes. All these numbers are so very important. Very good. Okay, yes, we all differ from 0.01, 0.1 kuda illa, 0.01%. Okay, yes. Coming to the last part, less than dash of the genome codes for protein. Less than dash of the genome codes for protein. Yes, this is interesting. Hi Sahana. Less than, very good, super. The numbers alla neenga marakave kudadu. Okay, dash make up very large portion of the human DNA. Yes, I am good. Neenga eppadi irkeenga? Dash make up very large portion of human genome. Repetitive sequence. This is what we are going to discuss next. Okay, repetitive sequence. Okay, dash are the stretches of DNA sequence that are repeated many hundred to thousand times. Dash are the sequence which are repeated many hundred to thousand times. What are they? Repetitive sequence. Ingiyo ade answer da. Okay, the same answer. Repetitive sequences are stretches of DNA, okay, that are repeated many times. Okay, some 100 to 1000 times. They do not code for any protein, okay, but they are thought to have shed, okay, or they are helping us to understand something. What is this? What are these three blanks? Anybody? They help to understand chromosome structure, okay, chromosome structure, evolution, and anybody? Chromosome structure, evolution, and one more thing is there. One more thing is there, dynamics. Okay, chromosome structure and dynamics. Okay, புரிஞ்சுதா? Yes, evolution ரொம்ப முக்கியம். உங்களுக்கு வந்து evolutionary significance அதிகமா இருக்கு. Yes, வெறும் less than 2% of my entire genome codes for proteins. Rest of them are all junk DNA. Okay, yes, let's talk about the next one. Chromosome 1 has dash and Y as the dash. Chromosome 1 has the most genes. Okay, chromosome Y has the fewest genes. Okay, yes. Scientists have identified about dash. இல்ல எவ்வளோ numbers இருக்கு பாருங்க. If you are good in remembering numbers, super, well and great. Okay, நீங்க numbers ஞாபகம் வச்சுக்கிறது கஷ்டம்னா டெய்லி இதை எழுதி பார்க்க வேண்டாம், எழுதினதை பாருங்க. Okay, yes. So என்னப்பா நடக்குதுங்க? Yeah, tell me. Chromosome 1 as 2968. Okay, and this is fewest எவ்வளோ இருக்கு? 231. Very good. Super. Yeah, so scientists have identified about 1.4 million. எவ்வளோ பசங்களா? 1.4 million locations where there are DNA differences in single nucleotide. ஒரே ஒரு nucleotide. இது ரொம்ப முக்கியம். This is very important. Okay, so DNA fingerprinting involves identifying difference in specific regions in DNA sequence called as dash. What do we call it as? DNA fingerprinting involves identifying difference in some specific region in DNA sequence called as. ஒரு specific name இருக்கு, அது பேர் என்ன? What is that name? We call it as variable. என்னன்னு சொல்றோம் பசங்களா? Variable number of tandem repeats. என்னன்னு சொல்றோம்? Tandem repeats ன்னு சொல்றோம். அது வந்து VNTRs ன்னு சொல்றோம். Okay, VNTRs called as repetitive DNA. Okay, one type of repetitive DNA is your VNTRs. Okay, so repetitive DNA. VNTRs are repetitive DNA va? Yeah, okay, inga under repetitive DNA. Okay, so VNTR is also right answer because VNTR is also a type of repetitive DNA. Okay, volume increase பண்ணவா? Okay, sure. அது என்னோட மைக்ல ஏதோ ஒரு சின்ன பிரச்சனை இருக்கு. Okay, I have to fix it. நான் ஆர்டர் பண்ணியிருக்கேன், மைக் வரணும். Okay, that's the thing here. Yeah, கண்டிப்பா இன்னொரு என்ன விஷயம்னா நான் இன்னும் கொஞ்சம் லவ்டா பேசணும். if I speak loudly maybe things will work better, right? இப்போ ஓகேவா? இப்போ என்னோட வாய்ஸ் ஓகேவா? Please do let me know. Is my voice okay now? Yeah, the bulk DNA forms a major peak and the other small peaks are referred to as dash. Simple, satellite DNA. அது என்னன்னு சொல்றோம் நம்ம? Satellite ன்னு சொல்றோம். Okay, I think now it's better, right? Now it's better. Is it okay now? Please do let me know. எனக்கே கொஞ்சம் பெட்டரா தெரியுது, கேக்குது என்னோட வாய்ஸ். Okay, MCQs and PYQs of reproductive health and sexual reproduction. Sure, கண்டிப்பா போடுறேன். Yeah, I know. Yes. So DNA polymorphism means, in simple terms, polymorphism variation at dash level arises due to dash. Okay, polymorphism variation at DNA level. எந்த லெவல்ல பசங்களா? DNA level arises due to mutation. அது எப்படி arise ஆகுது? Mutation நால arise ஆகுது. Okay, the technique of DNA fingerprinting was initially developed by, who developed it பசங்களா? Alec Jeffreys. எல்லாருக்குமே தெரியும். Alec Jeffreys was the one who basically introduced this technique. Okay, he used satellite DNA as dash that shows very high degree of polymorphism. It was called VNTR. What did he use this satellite DNA as? Anybody? Yes, Alec Jeffreys. மறந்துடாதீங்க இந்த பேரை மட்டும் மறந்துடாதீங்க. Okay, yes, DNA as dash. What is this? Ma'am, 12th board next class, ma'am. புரியல என்ன சொல்றீங்கன்னு. Okay, so he used satellite DNA as a probe. Probe na enna? Labeled DNA. Okay, yes. So that's it pasangla. I've covered all the possible lines which could be asked as a question. ஓகே எஸ் ப்ரோப்னா என்ன பசங்களா லேபிள்டு டிஎன்ஏ தான் நம்ம வந்து ப்ரோப்னு சொல்றோம் ஓகே சூப்பர் ஓகே வெரி குட் யா so any doubts in this session next session என்ன வேணும் நாளைக்கு என்ன செஷன் வேணும் நீங்களே டிசைட் பண்ணுங்க please do let me know you want MCQs you want one shot you want week quiz என்ன வேணும்னு சொல்லுங்க we will do the same okay so இப்போ எப்படி இருக்கு confidence how is the confidence in molecular basis of inheritance next section for 12th board class okay super molecular basis menti quiz okay MCQs okay fine MCQs பண்ணலாம் okay நாளைக்கு MCQs பண்ணலாம் நாளைக்கு காலையில 𝑣𝒆𝒄𝒌 தமிழ்ல I'm coming live I'm teaching sexual reproduction in flowering plants and evolution one shot okay so if you guys are free you can come and attend yes infinity super successfully revised yes polymorphism na there are little changes okay inge ponga yeah inge irukku parunga it means there are simple changes variation okay ஒரு nucleotide la variation irukkuradhu ரெண்டு nucleotide la variation irukkuradhu adha nama polymorphism nu solrom நீங்களும் நானும் ஏன் ஒரே மாதிரி இல்ல அந்த 0.01% difference ஏன் இருக்கு that is because of polymorphism okay super very good I hope all of you have enjoyed this session நாளைக்கு செஷன்ல molecular basis of inheritance menti quiz na podala we will discuss MCQs okay நிறைய MCQs கொண்டு வர MCQs discuss பண்ணலாம் yes kandipa see coming to your 𝑣𝒆𝒄𝒌 Tamil we are planning something very interesting so Friday வந்து நாளைக்கு ஃப்ரைடேவா சாட்டர்டே போர்ஷன்ஸ் முடிச்சிருவோம் சாட்டர்டே ஈவினிங்ல இருந்து உங்களுக்கு MCQs PYQs ஓகே மென்டி குவிஸ் அந்த மாதிரி எல்லாமே நம்ம டிஸ்கஸ் பண்ண போறோம் till Tuesday morning okay difference between intron and exon attach பண்ணலையா கண்டிப்பா சொல்றேன் okay ecology MCQs ஓகே என்ன MCQ வேணுமோ தயவு செஞ்சு chat section கமெண்ட்ல போடுங்க யாருக்கு அதிகமான கமெண்ட் வந்திருக்கோ அதை வந்து நான் வந்து நாளைக்கு பண்றேன் ஓகே I am ready with anything okay yes introns na enna they are non coding okay exons na enna they are coding okay they are a part of your hnRNA heterogeneous RNA so heterogeneous RNA la non coding and coding regions இருக்கும் அதை தான் நம்ம introns and exons னு சொல்றோம் yes சிம்பிளா நம்ம ரிவைஸ் பண்ணிட்டு இருக்கோம் ரொம்ப டென்ஷன் ஆகாம ரொம்ப ரிலாக்ஸா இதே மாதிரி படிச்சிட்டு போங்க we will see who will stop you from getting 360 out of 360 okay yes okay பசங்களா any other doubts high weightage கண்டிப்பா ரொம்ப high weightage உங்க நீட்லயும் சரி உங்க போர்டு எக்ஸாம்லயும் சரி this has very high weightage okay உங்க நீட் எக்ஸாம்ல கிட்டத்தட்ட 32 மார்க்ஸ்ல இருந்து 40 மார்க்ஸ் கூட வரலாம் இந்த வருஷம் தெரியல ஓகே அதே மாதிரி உங்க போர்டு எக்ஸாம்ல 10 மார்க் வரலாம் scientists name are important yes when it comes to NEET it is important boards I'm not very sure but for NEET it is absolutely important okay yes நாளைக்கு என்ன சாப்டர் MCQ சோ எந்த சாப்டருக்கு அதிகமான பேர் வந்து கமெண்ட் செக்ஷன்ல போடுறீங்களோ அந்த சாப்டருக்கான ஒரு 100 MCQs கொண்டு வரேன் we will solve it yes sure thank you so much take care pasangala take care bye we will meet morning sharp 6 am okay sexual reproduction in flowering plants and இன்னொரு சாப்டர் என்ன evolution okay yes ரெண்டுமே 1 hour தான் ஃப்ரீயா இருந்தீங்கன்னா காலையில எந்திரிச்சீங்கன்னா please do come and join the live take care bye biotechnology முடிச்சாச்சு நீங்க செக் பண்ணலாம் okay animal kingdom examples sure take care bye பசங்களா